Welcome to this year's Stem Cell Program event. I hope this finds you and your families healthy, certainly under these uncertain times. Thank you for taking the time to participate this year. Although we had to go virtual, we're happy to see so many of you logging in today, and it's really a great lineup. So I think you're going to really enjoy our speakers. This is actually the fifth time that we've offered this type of event. So let me provide you with a brief overview of the agenda. Dr. Paula Arlotto, our keynote speaker, will be followed by authors of the 3 highest rated abstracts selected by a group of stem cell researchers in our community. The winners are Wei-Chen Yuan from the Camargo Lab, Neha Nagpal from the Agarwal Lab, and Nan Liu from the Orkin Lab. For these efforts, each of the abstract winners will receive a $100 Amazon gift card, so congratulations to them. A link to today's agenda has been posted to the chat for your reference. Our session is expected to continue until approximately 4 p.m. And immediately following, we're actually having a special event, which is a birthday event for Dean George Daly. That Zoom link is posted in the chat room right now, and we'd be happy to have you join us. But first, we have a very special guest joining us today. As many of you know, Sandy Fenwick, Boston Children's Hospital current Chief Executive Officer, is retiring at the end of March. We're very happy to have our next CEO, Dr. Kevin Churchwell, take us off on the day. And Dr. Churchwell is currently the President and Chief Operating Officer of the hospital. He joined Children's in 2013 after coming back, I should say, as Executive Vice President for Health Affairs and Chief Operating Officer. He's an Associate Professor of Anesthesia at Harvard Medical School. Dr. Churchwell is a great friend of the Stem Cell Program. We really appreciate what he's done for us, and we're thrilled to have him here today. So I'll turn it over to Kevin.
Lynn, thank you. It's really exciting to be here today. And I want to thank everybody for attending this great day of community and collaboration. You know, today we celebrate the research being conducted every day in the Stem Cell Program and their affiliated labs across the children's community. At Boston Children's Hospital, you know, we are always pushing the boundaries of what's possible through clinical research and medical breakthroughs that save lives and transform how healthcare is delivered, both here and around the world. Boston Children's is the world's largest research enterprise at a pediatric institution. I think it's one of the largest actually around the world, no matter if it's a pediatric or adult institution, with more than 3,000 researchers and support staff and more NIH funding than any other pediatric medical center. The goal of the program is twofold: to develop an in-depth understanding of stem cell regulation and development, and from that understanding to derive stem cell therapies for treating both pediatric and adult diseases. The program continues to contribute to the mission of Boston Children's. Its leadership has established a strong base infrastructure with an outstanding core faculty. Additionally, they've created an active community of affiliated researchers and collaborators and host annual events, such as this seminar today. They've established a business-based model for the translation of its research into the clinic and continue strong mentorship of its postdoctoral research fellows and graduate students. Many previous Stem Cell Program members presently hold academic appointments at highly regarded universities such as Harvard, Cornell, NYU, Johns Hopkins, and UCSF. Others have advanced to impressive positions in industry, such as companies as Novartis, AstraZeneca, Bluebird, and Biogen. It's my pleasure to introduce our keynote speaker, Dr. Paula Arlotto. Dr. Arlotto is the Gallup University Professor and Chair of the Department of Stem Cell and Regenerative Biology at Harvard University. She is the recipient of many awards, including the 2017 George Ledley Prize from Harvard, the Fanny Cox Prize for Excellence in Science Teaching, the 2018 Friedrich Wilhelm Bessel Research Award, from the Humboldt Foundation and a 2019 Harvard professorship. So I'll turn it over to Paula for her presentation. Thank you.
Thank you so much, Kevin and Len, for inviting me. It's really a pleasure to be here today. The only thing that would make it better, actually two things. One, if Len had played the trumpet. Two, if we would be in person, but I hope that will happen. That will happen soon. Thanks so much for having me. I'm gonna share my screen. Can you see it?
Yep. Perfect.
Okay, great. So my lab has been interested for many years in sort of understanding how during development in utero, in the embryo, the brain is made. How are the different cell types of the brain produced? How do they come together to interact with each other to form higher order structure? How do they form circuit that then control brain function?
Thank you, Paula. That was fantastic. All right, so we'll get started with the abstract part of our session. And so it is my pleasure to introduce actually Wei-Chen Yuan, who's actually a member of my lab. Wei-Chen graduated from National Taiwan University, and where she did some really amazing work in terms of studying ubiquitin ligases in cancer. And she's been in my lab for about 5 years, and she's been interested in understanding liver growth. And her work is mainly based on discovering a new platform for doing in vivo CRISPR screens in the liver. And today she'll tell you about a brand new story, which is where she's identified an epigenetic regulator that controls liver cell plasticity.
Today, I would like to talk about the role of the HBO1 in hepatocyte to ductal cells reprogramming.
So our next speaker is Dr. Neha Nagpal from the Agarwal Lab. She received her PhD in biochemical engineering and biotechnology from the Indian Institute of Technology in Delhi, India, where she investigated the role of microRNAs in breast cancer pathogenesis. She joined the Agarwal at BCH in July of 2016 and has been developing small molecule strategies to modulate human telomerase. Specifically, she's discovered some novel small molecules that have the potential to restore telomerase maintenance in patients with telomere diseases and associated rare degenerative disorders. So the title of her talk is a bit of a mouthful. It's repurposing dihydroxyquinolisines to restore telomeres in patient stem cells. And as a reminder, please post your questions in the Q&A. You can do that throughout the talk or at the end. And Neha, if you're all ready to go, you can take it away.
Thanks, Trista. Good afternoon, everyone. Thanks for providing me this opportunity. Today, I'm going to present my work on repurposing DHQ molecules to restore telomerase in patient stem cells. Firstly, a brief introduction about the disease we are working on. Dyscrasia congenita is the systemic degenerative disorder characterized with high rates of bone marrow failure and myeloid malignancy. The disease presents itself in 3 typical features shown here. With the age, the cells of high replicative capacity, such as stem cells, they lose their self-renewal ability and hence the bone marrow failure. Genetic discoveries in the past 20 years have revealed it to be one of the telomere biology disorders. Telomeres are ends of eukaryotic chromosomes that need specialized multiprotein complexes shown here in color to maintain these ends. Central to these is telomerase complex, which by reverse transcription maintains these ends. Any functional impairment in the components of this complex, including the reverse transcriptase itself, its RNA component, and factors associated with its assembly and stability are known to be mutated in DC and other telomere biology disorders. Despite the therapeutic advances, there are no curative therapies for the disease. Hence, there is an unmet clinical need for the systemic therapy for this disease. Restoring TUT in these patients would be curative. However, ectopic expression of TUT risks cellular immortalization. For this and various other reasons, we decided to focus on its RNA component TUC which is the non-coding RNA, but it's a limiting component for telomerase activity in human cells. Shown here is a nascent TERK molecule, which exists in an extended and adenylated form. Once transcribed, it gets repaired by another enzyme known as PAN, poly-A ribonuclease. This enzyme removes or repairs the 3' end of the TERK, hence TERK gets stabilized and telomeres are maintained. By studying the DC patients, we found that another enzyme, PAPD5, is responsible for the adenylation of this TERK 3'. Once TERK gets adenylated, it gets recognized by RNA exosome, which recognizes and degrades the same. When PAN is there, it repairs it, but when PAN is mutated in case of the DC patients, the whole balance shifts in the favor of RNA exosome. Thus, it's the balance between the PAN and exosome that decides the fate of the nascent TERK. We hypothesized that if somehow we prevent this A tagging, we might restore the balance in the favor of TERK stability over degradation. To support this hypothesis, we knocked out PAPD5 in PAN mutant DC patient iPSCs via a CRISPR-Cas9 mediated PAPD5 genome editing and found that PAPD5 knockouts leads to increase in TERK accumulation as well as increase in telomere length. Shown here are the clones with no disruption to complete disruption of PAPD5 protein. Here are the mature TERK levels in these clones. As you can see here, the TERK levels, which are quite low in the patients, significant TERK accumulation occurs when we knocked out PAPD5. And importantly for telomere length, which is quite low in the patients, increase in telomere length can be seen when we knock out PAPD5. This suggested a therapeutic strategy to us, as PAPD5 knockout is well tolerated in the human cells and leads to the accumulation of TERK, the telomerase RNA component, the limiting component for telomerase activity. We decided to look for the pharmacological inhibitors of PAPD5. Recently, Roche, the company, they have discovered dihydroquinolizinone molecules, which in cellular assays suppress hepatitis B viral surface antigen. Further, a subsequent report has revealed that PAPD5 and a related enzyme, PAPD7, are the cellular host factors for these dihydroquinolizinone or DHQ compounds. As PAPD5 was the target of action, we decided to test these compounds and designed and performed a cell-based screen of dihydroquinolizinone molecules to test for their effect on telomere maintenance. To do the same, we synthesized 14 different DHQ molecules. Shown here are the chemical structure of these molecules. First of all, we tested these molecules for their target binding, and we generated recombinant PAPD5 for this purpose and tested the activity of these DHQ compounds with the recombinant PAPD5 via differential scanning fluorimetry. And as you can see here, compared to that of the DMSO as a control, all these DHQ molecules, they bind with PAPD5 in a dose-dependent manner. Next, we use these DHQ compounds and tested their effect on telomere maintenance. And for this, we used PAN-deficient patient iPSCs, treated these cells continuously for 3 weeks in the presence of these DHQ molecules. And as you can see here, compared to that of the DMSO as a control, increase in telomere length can be seen in all the different DHQ molecules. Thus, our DHQ molecules are the tool compounds that can be used for telomere therapeutics. This also suggests that these molecules or PAPD5 inhibition, this suggests the convergence of role of PAPD5 in hepatitis B RNA virus regulation as well as telomerase RNA regulation. For further in-depth studies, we decided to focus on the clinical candidate RG7834. And next we asked the question, can we use these compounds to restore telomere maintenance in a disease-relevant human stem cell model? And for this, we used peripheral blood mobilized CD34 positive cells from healthy donors. We modeled our disease in these cells via CRISPR of the telomere-associated genes. These edited cells were then infused in immunodeficient mice. And these mice were then subjected to treatment with DHQ molecules or the DMSO as a vehicle control for 6 weeks in their drinking water. After 6 weeks, the bone marrow of these mice were harvested, and we looked for the effect on TUG maturation and telomere length. Shown here is the effect on TUG 3' profile, and here you can see in case of AAVS as a control, the TUG exists in a mature form, while in case of PAN-targeted mice, the aberrant or extended TUG forms can be seen, which gets rescued when we treated these mice with RG7834 in their drinking water. This was further confirmed by deep sequencing of the TUG 3' ends, and as you see here, in case of the PAN-targeted mice, the aberrant or oligoadenilated TUG can be seen, which gets restored back to the mature form with the treatment with RG7834 or DHQ molecule. Thus, treatment with DHQ molecule or PAPD5 inhibition promotes TUG 3' end maturation in human cells in vivo. We next looked for the effect of these DHQ molecules on telomere length, and for this we performed flow cytometry-based fluorescence in situ hybridization. As Paula said, mice and human brains are different. Mice and human telomere lengths are also different, and because there is a huge difference in the telomere lengths of mice and human, we were able to distinguish the signal. Now the question was, what happens to the when we treated these mice with the drug? As you see here, compared to that of the control, when we targeted these mice with PAN, decrease in signal or decrease in telomere length can be seen, which gets rescued when we treated these mice with the DHQ molecule or RG7834. This can be further seen here. In PAN targeting was rescued with the inhibitor treatment. Overall, we can say that oral treatment of small molecule DHQ compounds rescue telomere homeostasis in xenotransplanted mice model of PAN deficient disease. To summarize, patient mutations have revealed non-coding RNA biogenesis pathway, and especially PAPD5 as a target for manipulated telomerase RNA. Hepatitis B surface antigen suppressor DHQ molecules inhibit PAPD5 to restore telomere maintenance in patient iPSCs. Oral administration of RG7834, a DHQ compound, rescues TUG and telomere length in human stem cells in vivo. In terms of its implications, we present first demonstration of restoring human telomerase via small molecule. Our strategy provides an opportunity to manipulate telomerase via TUG, the non-coding RNA component, rather than TUT, the reverse transcriptase, and this addresses many safety concerns. We present target validation in a disease-relevant human stem cell-derived model. In terms of our ongoing future directions to advance this work, via a high-throughput screen, we have identified a novel class of PAPD5 inhibitor, which is quinoline derivatives. These are the molecules we repurposed, and these are the quinoline derivatives that we discovered as PAPD5 inhibitors as a novel class. We are trying to develop and characterize these molecules for their role in telomere therapeutics. As the structure of PAPD5 is not known, via creating various deletion mutants, we have now defined the catalytic core of PAPD5, which is not only more stable, but also is more sensitive to the inhibition by the DHQ molecule or PAPD5 inhibition. Via using this catalytic core, we have generated crystal balls of this deletion construct, and we are now in the process of refining the crystal conditions. We presented a xenotransplantation, and we are in the process of developing an animal model via using non-human primates. And towards this aim, we have used rhesus macaque bone marrow stem cells, and we model our disease by targeting PAN in these, and the phenotype observed of aberrant TUG is responsive to RG7834, as you can see by the phenotypic reversal in response to the RG7834 or the DHQ treatment. Based on my data, we are actually trying to translate this into human trial, and towards this aim, one of our patients, which is critically ill these days, as because of the multiple organ damage, shown here is his lung scan, you can see the damage. And we used his bone marrow mononuclear cells. And as you can see here, the phenotype reversal can be seen in response to the DHQ molecules. Talks have been initiated for the approval from FDA and IRB to start the clinical trial for this particular patient. Hopefully, we will be able to save a life and many more in the long run. Finally, I would like to thank all the Agarwal lab members, especially Sunit, for his great mentorship. I'm thankful to all of our collaborators and funding agencies. I'm grateful to all of you for being here and selection committee members for providing me this opportunity. And also happy Diwali to all of you. May you know the bliss in this festival of night. Now I would like to take questions. Thank you.
Perfect. Thank you so much. That was a great talk. If people have questions, if you could please put them in the chat. While we're waiting, I'll just ask a quick one, and I apologize if I missed this at the beginning, but are there mouse models sort of with a phenotypic telomere disease phenotype that you could also test the drugs in, or do you think it's specific to the human PAPD5?
There are mice models mainly which are the major model that we can use is the TUG deficient mice, where mice TUG mTOG minus, and but in that case TUG is not there, and TUG is our target. Okay. So we are limited by that.
Yeah, I think it'd be lovely to see, you know, a reversal of the whole phenotype, but it's really excellent. Other questions for Nia?
I just have two quick questions. So first of all, what happens if you give your drug to normal cells? Do you actually increase telomere length in normal cells, or do they have to have this particular mutation?
That's a good question. When we give it to normal cells, we don't see increase in telomere length because in the normal cells, TERT is already there. So when the TERT is less, it rescues TERT. So our target is TERT. TERT has to be depleted so that we can increase it. So in the normal cells, we don't increase the telomere length. It's a rescue-based strategy where we are rescuing it, not increasing it.
That's cool. And second, I imagine that the vision for a potential clinical trials, I mean, I think this have to be a chronic treatment, I would imagine, right? Because as soon as you remove the drug, then the TERT would also be degraded again.
That's what we are expecting. That's a great question. And as the patient is already critically ill, so whatever we can try, that's what we are hoping for. If we can even like improve his condition, that would be better.
Thank you.
Great. Any other questions? Either live questions or in the chat? Okay. Well, thank you very much. That was a really great talk. Very exciting stuff. I hope the trial goes well.
Thank you very much. Thank you.
And I will hand it over to Ryan. Are you ready to go?
Yep, I'm here.
Perfect.
Thanks. Yeah, well, thanks everyone for the talks so far. I'm going to introduce Dan Liu. Dr. Liu completed his PhD training in the National Institute of Biological Sciences in Beijing in China, where he studied mechanisms of epigenetic inheritance. He recently joined the Orkin Lab here at BCH in 2017, where his main focus is to study how fetal hemoglobin, where he studies the fetal hemoglobin repressor, BCL11A, and how it controls the gamma globin repression. Dr. Liu is also a K99 R00 Career Development Award grantee, which is very exciting. Again, as for everyone, if you have questions during the talk, please put them in the question and answer section of the Zoom program. And the title of Dr. Liu's talk today will be a transcription factor competition model for hemoglobin switching. And I'll hand it over to him now and can start.
Hey, thanks, Ryan, for introduction, and welcome to Boston Children's Hospital.
Thanks.
So, yeah, our lab studied the mechanism of hemoglobin switching, and there are 3 beta-like hemoglobin genes in the human genome: embryo, a duplicated gamma gene, and a beta gene. These 3 genes are sequentially expressed during human development, from epsilon to gamma, finally to beta. So the switch from gamma in fetal stage to beta in adult stage has been a paradigm for studying developmental gene control for decades. However, the critical factor that repress gamma globin gene remained a mystery until about 10 years ago. A series of work from our lab demonstrated that BCL11A is a major repressor for gamma globin. At the time I joined Orkin Lab, what is known about BCL11A is that it is one of the two major repressors of gamma globin, and it can act as a therapeutic target for reactivation of fetal hemoglobin for the treatment of sickle cell disease and beta thalassemia, which are caused by mutation in the adult beta gene. The mechanism that BCL11A repress gamma involve the recruitment of NERD co-repressor complex, as well as long-range chromatin interactions. However, there are many critical mechanistic questions that remain unsolved, such as including where BCL11A acts to repress gamma globin. And upon depletion of BCL11A, what activates gamma globin expression? And how does BCL11A counteract transcription activator to initiate the repression? And these questions are the main focus of my study in the Orkin Lab. So to study where BCL11A acts, we first try to map chromatin occupancy of BCL11A through ChIP-seq, but experiments failed. So we turned to a novel method called cut and run. In this method, we incubate the BCL11A antibody with native nuclei, and then the antibody can tether a protein A micrococoa nucleus molecule to the BCL11A binding site. Upon activation of the nucleus, it can cut around BCL11A binding site and release the BCL11A DNA complex. And we will collect the DNA, prepare a sequencing library, and we can map where BCL11A binds. So this method offers many potential benefits when compared to ChIP-seq. It affords much higher resolution and allows digital footprinting, which I will introduce later. Using BCL11A cut-and-run in adult erythroid cells, we successfully mapped the BCL11A binding in the cell. And in these peaks, we identified a novel motif TGCC sequence. We are actually quite excited to see this sequence because the TGCC sequence appeared twice in the gamma globin promoter, and more importantly, the distal one of the motif have mutation that reported more than 30 years ago that are known to cause a syndrome named HPFH. And in this individual, they have persistent expression of gamma globin gene in adult cells, indicating that the distal element is a repressor that repress gamma globin. At the same time, we observed that both of the TGCC sequence overlap with a cat box. Cat box is a ubiquitous transcription activator elements, and the major TF that binds to cat box is NFY. So we want to ask whether BCL11A and NFY binds to these motifs, and how do they carry out the opposite function. So the BCL11A cut and run data shows that the BCL11A indeed bind directly to the promoter of the gamma globin gene, HBG1 and HBG2. If we zoom in, we can see that the peak are located right on the two duplicated motifs and with a bias to the distal motif. However, NFY only binds to gamma globin promoter after deletion of BCL11A, but not in wild type adult cells, suggesting that BCL11A may inhibit the binding and function of NFY. If you look at the zoomed in view of the peak, NFY also binds directly to the duplicated motifs, but with a bias to the proximal one. So are we able to tell exactly which motif do these factors bind? To answer this, we devised a digital footprinting assay using cut and run. Basically, cut and run is a nucleus-based method. The DNA that is bound by BCL11A should be protected from nucleus digestion. So if we count the nucleus digest frequency for all the bases around BCL11A binding site, we should be able to observe a value shift pattern. And the middle part is the footprint left by BCL11A. And this is indeed what we observed for B-cell LMA footprinting. And we can see a very good protection of TGA-CCA sequence by B-cell LMA in the footprint assay. We also observe a beautiful footprint pattern for NFY, but in this case, the flanking sequence of NFY is protected from digestion because the NFY DNA complex exhibit a nucleosome structure that can protect a longer stretch of flanking sequence. A really powerful application of Catamaran footprinting is that we can look at the footprint at a single locus because the method has very high signal to noise ratio. And here shows a footprint, single locus footprint of B-cell LMA at the gamma globin promoter with the distal and proximal motif highlighted. Now we can readily detect 3 footprints in this region. One of the footprints locates right on the distal TGA-CCA sequence, and another one overlap with the proximal TGA-CCA. And we calculate the binding probability of B-cell LMA based on the footprint strength, which reflects the probability of B-cell LMA binding at the motif. And we can see that B-cell LMA prefers the distal one of the 2 motifs. On contrary, NFY footprint shows that it binds to the proximal motif as shown by the protection of the flanking sequence of the proximal CCAT, but we do not see a protection at the distal motif, which is supported by the log odds ratio. So our conclusion is that B-cell LMA prefers the distal TGA-CCA motif, whereas NFY binds to the proximal CCAT motif in the gamma globin promoter. To functionally validate the protein binding, we performed base editing of these 2 motifs. Base editing of the distal motif leads to a dramatic increase of gamma globin expression in all the single cell clones analyzed. And this result also support the use of base editing of this motif as a therapeutic strategy to reactivate gamma globin gene and treat sickle cell disease and beta thalassemia. And on the contrary, after we base edit the proximal CCAT motif, we observe decrease of gamma globin expression in most of the clones analyzed. This result established the repressor role for the distal TGA-CCA and the activator role for the proximal CCAT sequence. And because of the very short distance between these 2 motifs, we asked whether there is steric hindrance between these 2 factors and whether that is the underlying mechanism for the hemoglobin switching. So to answer this, we used the dCas9 to mimic B-cell LMA function. In this experiment, as a control, we remove the B-cell LMA from the cell, and NFY binds to gamma globin promoter and activate gamma globin expression. And then we put the dCas9 molecule to the B-cell LMA binding motif. And we observe that NFY binding is evicted by the dCas9 binding at a distance, and gamma globin expression is reduced. Because dCas9 is not a repressor or co-repressor, so this action is probably due to a steric hindrance of these 2 proteins. And as a positive control, if we put dCas9 directly at NFY motif, NFY is also evicted and the gamma globin expression is reduced. And this suggests that the dCas9 is able to mimic the B-cell LMA function without a repressor function, but possibly through steric hindrance. So based on this evidence, we provide a simplified model for hemoglobin switching. So in erythroid cells that are at fetal stage or without B-cell LMA, NFY combines to the gamma promoter, activate the gamma globin expression through the proximal CCAT motif. But in adult stage cells where B-cell LMA is expressed, it combines through the distal TGA-CCA motif, evict NFY binding, and repress gamma globin expression to achieve hemoglobin switching in adult cells. And we also showed that dCas9 is sufficient to evict NFY and silence gamma globin expression, even if it's not a repressor. Finally, I'd like to thank my mentor, Stuart Orkin, for his great guidance and mentorship, and also all the collaborators from Stu's lab, JC, Dan, and Lucas lab, as well as collaborators across the world for their great work. And I'm happy to take questions.
Great. Thanks for the really nice talk. I have a couple that I was just thinking about that I can start off with. The base editing was a super cool, really nice data there. I'm wondering, what is the molecular weight of BCL11A versus Cas9?
BCL11A is about 100 kilodalton. Cas9 is a bit larger. It's around 150.
Yeah.
Yeah.
I'm just thinking like molecularly or like atomically. How do you imagine Cas9? I mean, Cas9 is a globular protein. It takes up only around the binding site that it binds, which is best case, even if you extend it a bit, it's best case 30 base pairs. So you needed to extend another 2x that distance in some direction to have a steric. So how do you imagine that happening? Like, what's the model in your head for how that works?
Yeah. So although the distance between these two motifs are around 20 base pair, actually I briefly mentioned that NFY actually occupies some upstream and downstream sequence and form a nucleosome-like structure. So it probably requires a stable interaction with its flanking DNA to bind to this motif. So they are practically, the distance between the two motifs are actually shorter than 20 base pair.
Got it. And also, just one follow-up. Oh yeah, go ahead. Go ahead.
Okay. Also, another thing I didn't mention in this talk is BCL11A probably form oligomers in vivo, which also make it bigger, bulky, so that it can facilitate the steric inverse.
Got it. And sorry, one follow-up to that is, so are there any notes about, is there any knowledge about affinity? So if you have both NFY and BCL11, why would you expect BCL11 to be able to compete off NFY? Especially if NFY is already there. You have the cell maturing, NFY is already expressed, presumably bound and expressing. Why would BCL11 come and kick it off?
That's a good question. We have in vitro measured affinity for both protein. Actually, NFY binds tighter than BCL11A, but we think the in vivo situation might be different. First, the chromatin environment will affect the binding of TFs to the chromatin. So the in vitro measured affinity might not determine what happens in vivo. The other thing is BCL11A has many cofactors to help establish a repressive environment at the gamma globin promoter. That includes another major repressor, LRF, which I also briefly mentioned in the introduction. The LRF, BCL11A, they both recruit transcriptional repressors, which can further facilitate the closing of the local chromatin, which may also restrict the binding of NFY. So
Got it.
Yeah.
Cool. I don't know if there are any other questions that I see. If not, I can thank Nan again and hand it over to Len.
Great. Well, that was a wonderful, wonderful day. I want to thank our keynote speaker again, Paula Urlata, for participating today. And I want to thank Wei Chen and Neha and Nan for just excellent, excellent presentations. Carla Kim, who chairs the Research Engagement Committee, asked me to remind everyone about Friends and Family Boston Trivia Night, which is going to be held on November 20th at 7 p.m. Prizes will be awarded to the first, second, and third place winners. Registration is required by children's members, and everyone is welcome to invite a guest. For more information, we've included a link to the Research Engagement Committee website on the chat. We also look forward to our next Stem Cell Day event, which we're planning for the fall of 2021, where we'll enjoy mini presentations of the stem cell research that's going all across Children's Hospital. And hopefully we'll be able to meet in person next year. Now, as I mentioned, you're welcome to attend a special birthday event for Dean George Daly, which will be at 4 p.m. on the nose. The Zoom link is posted in the chat, and I really hope you'll enjoy us. So I hope everyone has a wonderful holiday season and be well. And thank you very much for this excellent day.